Hello friends, today we are starting the new chapter, that is the cell division. Okay, the chapter is related with the cell division process. You have already studied the cell division, okay, into the 10th classes, okay, in the chapter that is life processes in living organisms. So again in the 11th standard there is a cell division is chapter. So here there are some more information. Okay, you will get some more information about the cell division, about the cell cycle. So we will start simply from the cell division. What is a cell division? Cell division means the process of formation of new cell from the pre-existing cell is called cell division. Means simply there is a formation of the new cells from the pre-existing cells. So this is pre-existing cell and this cell will produces two cells. Again this cell will produces two cells. Okay, number of cells. So each and every cell arises from the pre-existing cell. We have studied the cell chapter, okay, structural organization of the cell, and in this chapter we have studied this cell theory, and in cell theory told that each cell arises from the pre-existing cell. Okay, so each and every cell in the world or in a living organism comes from the already existing cell. So how it comes? So due to the process of cell division, the single celled stage of the humans, okay, the single celled stage of the human is the zygote. Okay, after the fusion of the sperm and ovum there is a formation of the diploid structure, that is the zygote, and zygote is a diploid structure. Okay, means it consists 46 chromosome. Haploid means which contains the 23 chromosome, half number of chromosome. Human sperm contains 23 chromosome, female's ovum contains 23 chromosome. As the fusion takes place there is a formation of the 46 chromosome, which is a diploid condition. So this single celled zygote start to divide, and due to this division there is a division of the cell, that is zygote is a cell. So due to division of the zygote there is a formation of the complete organism. So we are formed from a single cell stage, that is the zygote. Okay, why? Because of the cell division. Due to the cell division process there is a formation of the complete organism. So one cell undergoes repeated mitotic divisions and produces a different organ, different tissues and the complete body of the organism. So that's why this process, that is life process, cell division is very important. Again cell division occurs in the injury region. Okay, when you got injured due to the falling or due to some reason, or there may be an accident, okay, there is a formation of a new skin on your injured tissue or at a injured region. So that is why repairing of the body is the main function of the cell division. Okay, cell division occurs inside the body. Okay, means that the internal organ, if any internal organ gets damaged, there is a cell division to repair that organ. So in our body every time there is a cell division is occurring. So that is why the definition of cell division is the process of formation of new cell. There are new cell formation from the pre-existing cell is called as a cell division. Okay, now there are basically two different types of the cell division, that is the mitosis and meiosis. So RNA and DNA ratio determines the type of cell division. Okay, how? Suppose if RNA to DNA ratio is more than 1, the cell will undergo mitosis. Okay, cell will undergo mitosis, and if RNA to DNA ratio is less than 1, then it will undergo meiosis. Okay, now here more than 1, it means that the RNA content is high and DNA content is low. Okay, that is why it will undergo mitosis process. And here RNA content is low and DNA content is high, so it will undergo meiosis, so it will produces 4 cell. Okay, now next is the types of cell division. So there are basically two types, that is direct and indirect type of cell division. Now what is direct type of cell division? This direct type of cell division is also called as a amitosis. Okay? Amitosis means there is a no mitosis. Now what is actually this? So direct type of cell division means nuclear material. Nuclear material means the DNA. Okay? And in viruses there may be a RNA. Okay? But except viruses there is a DNA in the all living organism which acts as a genetic material, that is nuclear material. Okay? So nuclear material, that is the DNA, divides directly without the appearance of chromosome. So what happens in direct type of cell division? There is a no appearance of the chromosome during the cell division. Okay? There is a directly division of the nuclear material. So there is no formation of the thick chromatin network. Okay? So and another one is, and without formation of the spindle fiber. You know that the spindle fibers, okay, which are arised from the centriole. We have seen the organelle from the chapter, that is structural organization of the cell, centriole. Centriole arises the spindle fibers. That spindle fibers helps in the separation of the chromosomes during the cell division. But here is a no formation of the spindle fiber, because there is a no formation of the chromosome. That's why there is a no formation of the spindle fiber. But such a direct type of cell division or amitosis is shown by the amoeba. Okay? Or mostly the prokaryotic type of cell division. Okay? Which shows the direct type of cell division. The binary pigeon in amoeba, you know that the binary pigeon in amoeba. So it is a type of amitosis. Binary pigeon is actually a direct type of cell division or a mitosis. So this is the direct type. Okay? Next one is the indirect. This is the main topic from this chapter. Okay? That is indirect type of cell division. So indirect type of cell division includes the two steps, that is mitosis and meiosis. There are two different types, that is mitosis and meiosis. So mitosis, what happens in mitosis? In mitosis there is a no no division of the chromosome. No division of chromosome means simply the mitosis will produces the two cells and the number of chromosome will not reduce. Suppose one cell have a 46 chromosome in the human body, then the cell which undergoes the mitosis process, it will produces two cells, and that two new cells also contains the 46 chromosomes. Okay? There is no reduce in the number of chromosome. But in meiosis there is a reduce in number of chromosome by the half number. Means, and meiosis produces the 4 cell from a single cell, that is the mitosis and meiosis. Mitosis occurs into the body cells, okay, and meiosis occurs into the gamete cells, gamete producing cells, that is the during the spermatogenesis, means during the sperm formation, and during the oogenesis in the female, that is during the ovum formation. These are the gamete. Sperm is the male gamete and ovum is the female gamete. So we will focus mainly on this mitosis and meiosis. So now next topic is the cell cycle. In this chapter we will going to study the cell cycle and cell division. So what is actually a cell cycle? Cell cycle means the cyclical changes or the cyclic changes in a cell by which it duplicates its content and finally divided into the two new cells, that is the cell cycle. So it is a cycle. Okay? It is a cycle. So this process is a repeated process. Okay? It runs in a cyclic manner. That is why it is a cell cycle. So what happens? Cell cycle means the cyclical changes in cell by which it duplicates its contents. Okay? It duplicate means double करने. Okay? Double करने. So it doubles its content, mainly the DNA, that is the genetic material, and the cell organelle also gets double, and due to this doubling there is a formation of the two separate cells. So that is the cell cycle. Definition of cell cycle, simple definition. Now this cell cycle have stages. So cell cycle is basically divided into the two phases, that is division phase and interphase. Okay? Division phase and interphase. So division phase is also called as M phase, which is called as mitotic phase or meiotic phase. That is why M phase. Now this division phase or mitotic or meiotic phase basically have again two substeps, okay, which includes the karyokinesis and cytokinesis. कॅरी ऑनचा मतलब कॅरी ऑन म्हणजे काय तुम्हाला माहिती आहे, कॅरी ऑन मीन्स द न्यूक्लियस. ठीक आहे. सो कॅरिओकायनेसिस, सो देर इज अ फॉर्मेशन और सिंथेसिस ऑफ न्यूक्लियस. दॅट process is called as a karyokinesis, and cytokinesis means there is a formation of cytoplasm. ठीक है. So karyokinesis includes the 4 stages, that is prophase, metaphase, anaphase and telophase. So we will going to study in detail prophase, metaphase, anaphase and telophase from this M phase. ठीक है. And cytokinesis means there is a cytoplasmic division. These prophase, metaphase, anaphase, telophase are common for mitosis and meiosis, but there is a difference in between these stages. We will study according to that stages. ठीक है. So basically division phase has a 2 phase, karyokinesis, cytokinesis. Karyokinesis again have 4 stages, that is prophase, metaphase, anaphase and telophase. तुम्ही शॉर्टकट मध्ये लक्षात ठेवू शकता PMAT. ठीक आहे. PMAT, prophase, metaphase, anaphase and telophase, that is PMAT. And next one is the interphase. Interphase, which is also called as I phase. It is a phase between 2 successive mitotic division. ठीक है. So now we will study the interphase first, and then we will start the division phase. Then in division phase, first is karyokinesis, and then after karyokinesis there is a division of cytoplasm, and then finally there is a complete formation of the 2 cells. So interphase basically this is the most active phase of cell cycle. In interphase the cell is the most active. ठीक है. We will study with the diagram. ठीक है. So here is a M phase. M phase means mitotic phase or meiotic phase, means it includes prophase, metaphase, anaphase and telophase. So this portion represents the division phase or M phase. ठीक है. And the remaining portion, this portion represents the I phase, means interphase. ठीक है. And this interphase again divided into the 3 sub-steps, that is G1 phase, S phase and G2 phase. So this is a complete cyclic manner. So cell division starts from G1 phase, then undergo S phase, G2 phase, then prophase, metaphase, anaphase, telophase, and then finally there is a formation of 2 new cells from a single cell. So this is the complete cyclical changes, or this is a complete cell cycle. So we are going to in detail in G1 phase, S phase, G2 phase, then the division phase, that is prophase, metaphase, anaphase and telophase. So this interphase, so this is the interphase. ठीक है. This interphase is also called a preparatory phase. Why this is called as a preparatory phase? Because cell prepare itself for the cell division, for the karyokinesis, that is for the mitotic phase, that is division phase. Cell itself prepare during the interphase for the division phase, that is why this is called as preparatory phase. So what preparation? So preparation is the synthesis of the DNA, ठीक है, then RNA molecules, proteins which are required into the division of the cell, ATP molecules for the energy, different types of enzymes which are required during the cell division process takes place in this phase, that is interphase. That is why interphase is called as a preparatory phase. Simple. Then this stage, that is the interphase, it covers more than 95% time of cell cycle. ठीक है. So more than 95% time is required for the interphase, and the M phase is a short period. So that is why in this diagram we are representing interphase. ठीक है. Mostly the larger portion is of interphase and smaller portion is of M phase, that is division phase. So almost 95% time is required for the preparation of the cell for the cell division, and then remaining 5% time is only required for the division of the chromosomes. ठीक है. So these interphase includes the total 3 steps, that is G1 phase, S phase and G2 phase. ठीक है. This G1 phase is called as a post-mitotic phase. Post-mitotic phase, because this G1 in this diagram present after the mitotic phase. Okay, that is why post-mitotic phase, and G2 phase is called as a pre-mitotic phase because it is present before the mitotic phase. That is why G2 phase is called as pre-mitotic phase and G1 phase is called as a post-mitotic phase. This point is very important. So many students confused with the pre-mitotic and post-mitotic. Okay, which stage is called as a pre-mitotic phase and which stage is called as a post-mitotic stage. So simply, if you learn this diagram, then you can easily visualize the G1 phase, S phase, G2 phase, and you can easily answer the question which is the pre-mitotic phase and which is the post-mitotic phase. Okay, G1 phase means growth 1 phase, S phase means synthetic phase, there is a synthesis of the DNA RNA molecules, and G2 phase is again a growth phase where the cell grows, and after fully growth of the cell undergoes the division phase step by step: prophase, metaphase, anaphase and telophase, and finally there is a formation of the 2 new cells. In mitosis there is a formation of 2 cells, and if the cell division is meiosis, there is a formation of 4 cells, and that 4 cells are haploid, means the chromosome numbers are half, okay, as compared to the parent cell. So here we completed the cell division definition and cell cycle, okay, and the interphase. Okay, we will see the G1 phase, S phase and G2 phase. Our next topic is the interphase. We have seen the basic about the interphase, which includes the 3 substeps, that is G1 phase, S phase and G2 phase. Now we are going to study in detail the G1 phase, S phase and G2 phase, that is the interphase. So what is basically the interphase? Interphase is a period between the 2 successive divisions, or it is a period between 2 successive mitotic divisions. So here is a diagram of cell division which shows the 2 different phases, that is interphase in blue color and mitotic phase in black color. We have seen the sub stages of the mitotic phase, that is prophase, metaphase, anaphase and telophase. So this portion represents the mitotic phase and this larger portion represents the interphase, which have G1 phase, S phase and G2 phase. So that is the complete cell cycle. So interphase is a period between the 2 division phase, 2 successive division. Okay, because interphase is not a division phase. Okay, mitotic phase is a division phase. So it is a period between 2 successive division. It means that between the 2 successive mitotic divisions. Now there is a first step, that is the G1 phase. Interphase is divided into the 3 sub stages, that is G1 phase, S phase and G2 phase. Okay, first one is G1 phase. Simply G1 phase means first growth phase, that is why it is represented as a G1, means the first growth phase. In this first growth phase, cell start to grow, cell enlarges. Okay, and this G1 phase, that is first growth phase, also called the post mitotic gap phase. Why it is called as post mitotic gap phase? Because of G1 phase is present after the mitotic phase, that is why it is a post mitotic gap phase. So it is a gap. Okay, interphase is actually a gap phase, so that is why it is called as post mitotic. Why post mitotic? Because after the mitotic phase is G1 phase, that is why post mitotic gap phase. Now what happens in this G1 phase? It is very important because it involves the synthesis of the proteins, mainly the enzymes which are required for the cell division, which are required for the DNA replication, and the energy molecules, that is the ATP molecules, are also synthesized into the G1 phase, which are required for the DNA replication. Next point, proliferation of cell organelles takes place. Now cell organelles like the mitochondria, Golgi body, endoplasmic reticulum, then chloroplast into the plants. So proliferation means the division of the cell organelles takes place into the G1 phase. And another point is the cell may withdraw from cell cycle and enter into the G0 phase. G0 phase is the phase in which the cell cycle is inactivated. So after the G1 phase, cell may enter into the another phase, that is the G0 phase. The cell in G0 phase may again re-enter into the G1 phase or may get differentiated and become the permanent cell. Means after entry into the G0 phase, the cell may differentiate and become a permanent cell. Means there is a differentiation of cell. Okay? Differentiation means the cell acquires the specific character, specific functions. Suppose there may be a heart cell which may differentiate into the heart cell, may be differentiate into the liver cell, may be differentiate into the kidney cell, may be differentiate into the brain cell. So this is the differentiation. So cell may re-enter into again the G1 phase, or it may get differentiated and become the permanent cell. Once it becomes the permanent cell, it does not differentiate. Okay, so that is why the G1 phase G0 phase is also important. Now what is actually the G0 phase? In G0 phase, cell is metabolically is active, means there is a catabolism and anabolism, and cell does not grow into the G0 phase and after also does not differentiate further. Once it differentiates, again it cannot differentiate. So another one, an important point about the G0 phase is the loss of control at the G0. When cell enters into the G0 phase, it is a temporary phase, okay, or it is also called as a rest phase. So when the cell enters into the G0 phase, and if there is a loss of control at the G0 which leads to the uncontrolled division, uncontrolled growth, as in tumor or the cancer. So you know that in the cancer basically there is uncontrolled growth of the cells. There are different types of the cancer. You know that the blood cancer means uncontrolled growth of the WBC. So it means that there is no control on the cell division process. That is why there is a uncontrolled division. Now this control is a genetically controlled. Okay, there is a genetic control. So when there is a loss of control at the G0 stage, it leads to the uncontrolled growth, and which leads to the formation of a mass which is called as a tumor, or that condition is called as a cancer. So another an important point in G1 phase is there is no duplication of DNA in G1 phase, so DNA content does not get double into the G1 phase. Okay, in G1 phase DNA content does not get double because in G1 phase there is no duplication or there is no replication of the DNA, but there is the formation of the energy molecules which are required for the DNA replication. So it means that there is no duplication of DNA in the G1 phase. Now the next phase, here we completed the G1 phase, first phase. Now the second phase, that is the S phase. This S phase is called as synthetic phase because there is a synthesis of the DNA molecule and histone proteins and another protein molecules. That is why it is called as a synthetic phase. So important point in S phase is the replication of DNA. Replication of DNA. Replication of DNA takes place into the S phase. That's why entire genetic material of the cell gets double. It means that if in the G1 phase the DNA content, suppose the DNA content is 2n, if the DNA content is 2n, it will become 4n, means it gets double, but the number of chromosome does not change. If the chromosome number are 2, after the S phase the chromosome number will be same, that is the 2. There is no increase in number of chromosome, there is only increase in the amount of DNA. Don't confuse with the increase in DNA and chromosome. There is only increase in the amount of DNA, but does not increase in number of chromosomes. Okay, so you have to remember this point because the questions may asked from this point. Okay, because it is little bit confused point. Now the nucleus size also increase in the S phase due to doubling of the genetic material. Means there is only replication. Here you can understand easily, there is only a replication of the DNA. Suppose this is a chromosome and inside chromosome is made up of the DNA. So suppose this is the DNA, this is the old DNA, okay, old DNA, and on this old strand there is a synthesis of new strand which is represented into the black. Okay, so now the amount of DNA is doubled but the chromosome is 1. Here number of chromosome is 1 but amount of DNA gets doubled. Okay, because there is no separation. After the separation of these chromatid, the number of chromosome will get double. And another one point, that is histone proteins are also produced into the S phase. These histone proteins are required for the packaging of the DNA, because the DNA gets doubled in this S phase. That's why histone proteins are also required for the packaging of that DNA. So this is a simple, that is the S phase, which is called as a synthetic phase, which is the second phase into the interphase. Now after the S phase there is a G2 phase. G2 phase means it is a second growth phase, that is why it is represented as G2 phase. Now the G2 phase is also called the pre-mitotic gap phase, because G2 phase is a present before the mitotic phase in the cell cycle. It is present before the mitotic phase in the cell cycle, that is why it is called as pre-mitotic gap phase. Now what happens into the G2 phase? It involves the synthesis of the tubulin. Now these tubulins are actually the microtubules which are required for the centriole formation. Then the synthesis of RNA and spindle proteins also takes place. Then duplication of cell organelles takes place into the G2 phase, and again there is a growth of the cell, that is why it is called as a growth phase, second growth phase. So duplication of cell organelles like the mitochondria, endoplasmic reticulum, Golgi bodies and the chloroplast into the plant cell. Because during the cell division, from one cell there is a formation of two cells. So that's why we also require the doubling of the organelles. That's why duplication of cell organelles takes place into the G2 phase. Next, centriole also duplicates. We have seen the centriole, the structure of the centriole into the cell structure and organization chapter. So the function of centriole is to separate the chromosome. It helps in the mitotic division and meiotic division to separate the chromosomes. So that's why initially in the cell it is present only one. Okay, in each cell it is one, but during the cell division it also gets duplicate, because for the division of chromosome two centrioles are required to pull the chromosome at the two opposite poles of the cell. So actually the cell prepare to enter the mitotic phase into this entire interphase. So actually this entire phase is actually a preparatory phase, means cell prepare itself to enter the mitotic phase, or it gets ready now for the division. For division we require a double amount of DNA, because from one cell there is a formation of two cells. So we require the genetic material in another cell also, and we also require the cell organelles in another cell. So that's why this cell is actually this phase, that is interphase, is actually a preparatory phase, and after the preparation the cell enters into the M phase, that is mitotic phase, and then starts the actual process of cell division. First step is the prophase, second one is metaphase, third one is anaphase, and fourth one is telophase. and after this karyokinesis, this is actually a karyokinesis, and after karyokinesis there is a cytokinesis, and then finally leads to the formation of two different cells which are exactly similar to each other, because this is a mitotic division, and mitotic division does not produce variation because there is no mixing of the genetic material, there is no recombination. So here we completed the interphase, which include the three stages, that is G1 phase, S phase and G2 phase.